care about, how much these things are. Also, the binomial Punnett square problem is also on the final, usually on the final, so that's 6 points. And also, you're gonna spend a lot of points on these things through the semester. It's worth sort of understanding how they work and how we do the problem. We left off here, which is kind of a weird place to leave off because we're right in the middle towards the end of the calculation, and you might not remember what we did. But we'll go over the rules and sort of steps in just a second. I'll wait until we sort of get through this part. We left off with this. Again, there wasn't a whole lot of time at the end of the last class, but why do we use 4 numbers in our numerator? And again, what you want to remember is we are calculating the distance between 2 genes. And because of that, we need to use every single recombination event that happened between those 2 genes. That's why we use the single recombinants giving us the 40 and the 30 offspring, but also the double recombination events giving us the 4 and the 6 offspring also have to be used. Because in that double recombination, the same crossing over event happened as in the one we're looking at. This is again one place where a student can very often make a mistake. It does not include the double recombinant and the single recombinants when they do this calculation. And 740 is just the total. So if you do the math, we get a map distance of 10.7 centimorgans. At this point, we have 6 of the 8 numbers already identified. So to do the next calculation should be fairly easy. It doesn't require any thought, except take the numbers you haven't used yet. So as long as you can spend all of your energy on that first decision, the second decision is already made for you. The other thing you should think about is, with the numbers give us some information on distance. Because the further things are apart, the greater or the more often recombination happens. You get a bigger recombination frequency because of a larger distance. And more recombination means more of those offspring showing up. So it should make sense that when we use 59 and 44, we get a bigger distance than when we use 33 and 40. If you get the wrong answer, or get a different answer, you should think about this. This doesn't make sense. More offspring should be more crossing over, which should mean larger distance. But here we get a distance of 14.7 centimorgans. Again, you are required to give me 3 distances, not just 2 distances. And so the last distance we would calculate would be the distance between the outside genes. And here we simply use the addition rule. Combine the 2 together, so we get a total distance between L and G of 25.4, 10.7 plus 14.7. The other reason why I ask you to do all 3 distances, but I only ask you to calculate 2 distances, is to give away what the middle gene was. Because it'd be the one that would be common between both. You probably don't care why I do things I do. Do you care why I do things I do? And try to hide the answer by making you calculate all 3 distances. Again, the third distance should be pretty easy. Just add up the 2 distances you had. You can see the other place you could potentially get wrong answers. If your answer starts with 0 point something, you probably want to reevaluate because that distance will be too short. Basically, these are the same gene, so always also integer sort of answers. It's anything above 1. Typically things above 5, or we typically see 10 is probably the sort of lowest that we typically see. Before we go into doing another example of this, there is one more potential thing to think about. What is that thing? Just kidding. What does the multiplication rules say? Who remembers what the multiplication rule says? How do we apply the multiplication rule, or when do we apply the multiplication rule? Under what what context do we apply the multiplication rule?
Is it when you say or?
No, or statements go with the addition rule.
Isn't it and then?
So if something says and then, we typically will use the multiplication rule. But why is and then in the multiplication rule together?
Probability that two things are gonna happen, so you have to do probability of one times probability of another.
And the other really important thing I think you're missing there is when we think about the multiplication rule, we assume that both events are independent of each other. One should have no bearing on the other, which is why we can treat them the way we do, which is simply combine them together with multiplication. If we do that with our probabilities, and the people have done this, should recombination or crossing over between L and GS have any impact on whether crossing over happens between S and G? You may think it's a trick question because we're talking about it. But if you just look at the first part, like why would one thing affect another thing? It turns out it does. And if you use the multiplication rule, we have the probability of crossing over between here is a single crossing over event. The probability here of crossing over. Combine them together, this is the probability of the double recombination event happening. And if we take this percentage and use the total number of offspring that we got from our question, or from our experiment, we should get 12 offspring, or about 12 offspring from the double recombination event. And we know that's 6. And people have seen this a lot. The expected number of offspring is not the number of offspring we always get. It's always much lower. And what is the explanation of this? It's an idea we call interference. Interference is kind of the reverse linkage idea. Linkage says that things are close to each other, close together, so recombination is rare between them. Interference says that recombination in point A prevents recombination from happening too close to itself. So recombination events are spread out. That's what interference does. It sort of spreads out the recombination events so they don't happen too close to each other. Does anybody have any ideas or thoughts of why we want to spread out those changes? Spread out those events, make recombination further apart rather than really close to each other. How much change do you get if things are really close to each other? So we take this picture. How much variety do you get if you have this happening versus this happening? That's the idea of interference. If the crossing overs are too close to each other, there's not a whole lot of variation that's being introduced. You get one difference. Who cares? One difference might not really matter. But if you get 5 or 6 differences, then you get a lot more different combinations and possibilities than you had originally. So we want to spread these out so the dots are close to each other so we get a little more variation happening, so more differences. And it's kind of hard to say more because it's the same number of differences, but not exactly the same number of differences. This is really just a definition, so as long as you understand that interference is what it is, it says that recombination events don't happen nearby each other. They're more likely to not occur nearby each other. So one happening sort of prevents another happening nearby. And maybe it's the actual machinery of the DNA coming together that sort of creates a bubble. That bubble prevents another formation of another structure too close. So they're going to be spread out because of the size of the structure that's made and all the things that go into changing the DNA in some way. Or maybe it's not, who knows? I don't know.
So does that increase just genetic diversity? Is that what the purpose of it?
I don't know if it, I'm gonna say yes, but I could argue the other way as well. Does it actually increase genetic diversity? bit more because when they're further apart, you're gonna put more than one gene phenotype difference. And so if this first event happened, then only this one trait got swapped. So everything else is the same except one thing. If we do the other one, then at least 4 traits are now swapped. And so we get a little bit more variety because 4 things get changed rather than just one thing gets changed. So each time we go through a round of meiosis, a new crossing over possibility happens. Again, double crossing over is a rare event, much more rare than a single recombination event. And so we just cross over once, we're getting like almost maybe half the things swapping positions. And we don't want to sort of go against, we don't go backwards. Get crossing over too close to each other, we're basically remaking the same thing. So we try to spread them out so we get more variety. I never asked you to explain it or sort of draw this idea, just interference. These crossing overs don't happen nearby each other. And we're gonna put our knowledge to the test. And these are the steps. And we have to deal with. In a lot of places to make mistakes, and hopefully I'll tell you where the mistakes are most likely going to occur. This is where we're going to make a mistake. A lot of people will simply identify the middle gene and use the letters in the alphabetical order in which they're given to you. That's going to give you the wrong distances sometimes. So make sure you do rewrite. And we'll try to do the wrong calculation today that proves to you that if you don't do it the right way, then you're going to get the wrong answers. And once you rewrite the things, then you can do your crossing overs and identify your offspring and do your calculation. Just make sure that when you do your calculation, you use both the single recombination and a double recombinant offspring when you do this. So let's go ahead and see how well we do as a class. You can work this on your own. We'll sort of jump through the hoops. There will be 3 of the 3-point cross problems on the lecture review for today. There will also be a PDF file that sort of walks you through the calculations for each of them. And then on Monday, we'll also do another one at the beginning of class. See how everybody is doing with them. So what I'm gonna do on Monday, I'm just gonna give you the data and say, calculate, take about 5 or 10 minutes, maybe 10 minutes, I guess. The other thing you need to do is you need to think about, okay, how long does it take to do this? Because you can't spend all your time on this because you have the rest of the exam to do. Now, sometimes the numbers are formatted like this. Sometimes they're just straight up and down, kind of like the other problem. This one kind of gives things away a little bit. Why does it give things away a little bit? Well, they're already paired together, and not the numbers, the letters. Remember, these letterings form pairs. The numbers can lie, the letters won't lie. So they're already in pairs. And that makes it a little bit easier to sort of think about things, is these are all sort of the opposite of the things we think about a heterozygous organism. That's one thing you kind of do to start with is sort of pair things together genetically and then start to think. But hopefully we can do the first part, which are our parents. And what information do we use to identify the parents? Why are the parents the parents?
They're the most often.
They are the offspring that show up the most often. And the reason they show up the most often is why?
Crossing over is rare.
And we assume that crossing over is rare. So our parents should be the things we get the most of. That means our parents are these two. And which of these numbers represents our double recombinant offspring? And why are they the smallest?
That's right.
Again, just trying to reinforce the idea that crossing over is rare, Double crossing over is the most rare thing, so we pick the ones with the lowest numbers, which are gonna be the 27 and 23 numbers, where you get A, B, E, and A. Some of you might look at these and say, oh, that's a diploid cell. It's not, they're individuals. The way this was really done was by making haploid cells. So we're looking at things individually, and doesn't an organism where it works. So sort of think about the actual logic of how it happens. We have our parents and we have our offspring, each with 3 genes. And now we move on to the next step, which is identifying the middle gene. And how do we identify the middle gene? What sort of rule defines what the middle gene is? I talked about this last time. The middle gene is the gene that's going to do what in a double crossing over event? Let's draw it out. And if we do a double crossing over event, what trades places? Well, overthink it, just look at it. Which thing is going to trade places? Yeah, the 2. The cardinal number and the non-cardinal number, or the Roman numer, I guess you call it. Those trade places. So in a double recombination event, you want to remember the middle gene is the thing that trades places. So because we know this fact, the middle gene is the gene that's going to swap places. The outside ones stay the same. The inside one is the one that changes. So we compare our parent, the one without crossing over, to the double recombinants. What letter has changed positions relative to what it started with? Okay, this is what we start with. Capital A, little b, big E. Which of these looks most like that? Meaning we have the same 2 letters, but a different third letter. Which of these 2 looks almost like this one here? And if we compare capital A, little b, capital A, little b, the E is the thing that's different. And we should assume, because the E is the different thing, E is in the middle. And we do the same thing here. We see the same result. Same A, same B, different E's. Indicating that E should be the middle gene. So both ways you do it, you should get the same answer. That E is in the middle. Now sometimes it'll be weird because the gene you're looking for is sort of, the B might be the one that changes for whatever reason, or the A, you never know. This one's a little easy because the letters are sort of look together. That's what we're trying to do. Trying to look for 2 the same, 1 that's different. Compare the parents to the double recombinant offspring. Now it's easy to make a mistake here if you don't use the right thing to compare to the right thing. As long as you compare the parents to the double recombinant offspring, you should immediately be able to identify the middle gene. So now we know the middle gene, we can rewrite our parents with that gene in the actual middle. And we're drawn like this because we want to represent that it is actually a diploid organism that's going to undergo the process of meiosis. And diploid organisms make haploid cells when we go through meiosis. So these are all haploid cells. You don't call them offspring. These are the haploid gametes that we make from meiosis, starting with these parents. So now we have the order and the order written correctly. We need to do our crossing over. We'll look at the crossing over between these first genes here, between the A gene and the E gene. And let's see what we get. So we'll do our next calculation, our first calculation. Combination frequency between gene 1 and gene 2. And I do recommend writing these things out. It'll make your life a whole lot easier. easy to get lost in the letters if you're not really paying attention. So if we do the crossing over between A and E, what are the products? Start with capital A, cross over, and now you have little e, continue on to get big B. Which number goes with this gene type? Right there, it's 128. When we do it here, let's start with little a, go down, cross over, get big E, continue on to get little b, which is now the 132 number. So these are the numbers we use when we do our first calculations. So we'll take 128 plus 132 plus 27 and 23. Because our double recombination event also included this crossing over between A and E. And divide by the total of all of these numbers. What is the total of all these numbers? They do the math faster than I can. Let's see.
110. Oh,
All 8 numbers. And make sure we use all of our offspring numbers, because using the total number of offspring. Do they have an answer yet? Definitely has a 2.
2009.
And it has a 4. And 29, 34. So we take 128 plus 132 plus 27 plus 23. We get 310. 310 divided by 2934. That's 1. A to E, E to D. So we know the distance between A and E? Yes. Nine.
10.6.
Did you get 310 divided by 2939?
I guess I put a number in wrong.
I got 10.6. Because when I added these 4 numbers, I get 310. 310 divided by that. Gave me
Yeah.
10.6.
I put 32 instead of 23.
And that's another sort of. There's a lot of numbers you gotta add up, so you wanna be quick, but not reckless. So the question is, who is completely lost? It's okay to be completely lost. First time we're doing it. Then it's the second time we're doing it. Yes.
I'm not completely lost. I just got a question. When you did the crossover, why did you only cross over the first 2 letters and not the last 2?
To calculate the distance between the A gene and the E gene,
Mm-hmm.
we use a combination between A and E.
So then you would just kind of essentially ignore the B because you're not worried about the B yet.
We're not worried about that part of the equation yet.
Okay.
We'll do the E to B crossing over to get the next distance.
Okay.
We don't have to worry about the double, doing the double, because we already know what the double numbers are. We can do the double and prove that when we do the double recombination event, we get these as our products, which is the number 27 and 23.
In the numerator, where did we get the 27 and the 23? Like, I understand for the 128 and the 132. But
What are the 27 and 23 offspring? They're the offspring that arose from a double recombination event. So double recombination event requires this to happen, but also another event to happen between E and B
Okay,
to get us this outcome.
okay.
But because to get a double recombination event, this also had to occur is why we include the single numbers and the double numbers together.
Okay,
Because the double contains the single in itself.
okay.
Any other burning questions?
I just think it was confusing me, where you drew the X, how that it recombines to be, it's just the uppercase and lowercase a switching places, not
No,
necessarily.
you follow the line through. This crossing numbers gonna swap things from one side to the other. So one of the easy way to do it is start on the left, go to the right, and follow your way through the different letters as you follow the line. So if we start here with our A, cross over and we go this direction. And the whole point of this is to reinforce the idea of what is crossing over actually doing. It's swapping letters. But yeah, you're right, you just swap the A's. But this is a sort of a conceptual way to see the crossing over happen and see the product. And some people do their heads are fine. But seeing like this sometimes reinforces, okay, well, yeah, these things are moving from one side to the other. Or picking up new partners. I wouldn't call it a square dance, but is it does do? Swing your marker around and round. Nobody's been to square dance before. Well, that's our first distance. What is our second distance going to be? We do the recombination event between the second gene and the third gene, which if we use our same drawing up here, we'll draw another crossing over event here. And we'll pick a different color for the new event to happen. So if we look at trying to calculate the distance between the second gene and the third gene, which is E and B, we go capital A, capital E, cross over to give capital D. A, E, B. And we start here with little a, continue on to get little E, cross over to get little B. We get little a, little E, little B. Which are these numbers here? We should already know that. We know these are the doubles. We know these are the parents. And we already said this is our first set of numbers. This has to be the second set of numbers. So we'll take 301 plus 308 plus 27 and 23. Divide by the total number of offspring in the experiment. And we get our second distance. Anybody have the answer yet? Well, I got 22.5 centimorgans. And if we want the distance between A and B, we just need to add up those two numbers together. Which would be 10.6. 31 point, 32 is on. And this thing I've drawn very times what I call the genetic map with our distances. Most times they don't make you draw a map because the problem just says part A is distance between 1 and 2. Part B is distance between 1 and 3. Part 3 is distance between 2 and 3. But what would we get if we did not rewrite our letters? if we use the original starting in alphabetical order, and we do our first crossing over between A and B, what genotypes do we get as a result? Just follow along. We get big A, big B, little e, and little a, little b, big E. What numbers do these go with?
128 and 132.
Alright, so that worked out okay. Maybe Dr. Pritch is full of shit, right? What numbers, what distance did we get when we used these two numbers? When we do a crossing over here, we're trying to calculate the distance between A and B. Does anybody see the problem? A and B is actually 33.1. But because we didn't rewrite them, we think the distance now is 10.6. You are correct, the distance between the first two genes was 10.6, but you have the wrong two genes that you're looking at. So that's why we need to rewrite them, because you may think you're calculating a distance for a distance you're not actually calculating.
Okay, so this is like a question about like how we found which one is the middle gene. Like you said that in a double recombinant, the middle gene changes position,
Yes.
but like when we have this one over here, it's like the E and the E. You said that that's the middle gene, but it hasn't flipped.
It did. E, cis or trans with A?
Cis.
Okay, right, because it's on the same BCDNA. Look right here. Is E still cis with A?
No.
And so the idea of crossing over is the A is no longer with the same E.
Okay.
Because now little a is with E, and big E is with E when these two were with E to start with. So in our doubles, we've swapped one of the genes, because that's the gene that's in the middle, we've changed that cis-trans configuration idea. Actually, I give you a choice today. We can either do another problem right now, or we're not leaving, because I'm gonna use the rest of the lecture. It's the question of, do you wanna work another problem, or would you rather me introduce the next topic?
Another problem.
You can go it out. Another problem. Let's find a problem from the list of problems. Not the answers. I'll give you the answers. Let's see. Where do I find? I think I'm gonna find it. It's not there. Let's go here. So here, here, over here, and here. Which test was it? Let's see. One square. Exam 2, okay. It's definitely, if you have bad eyesight, I apologize. And also finding the way, I apologize as well. Individually or as a class together? These folks, individually or as a class together? Together or alone? Together or alone? Please somebody say something.
Together.
Together or alone? Okay, together. What's the first step?
Find the parents.
Step number 1, parents. What are our parents? And again, why are they the parents? Reinforce everything. They're the most common, but why are they the most common? Crossing was rare. The most common ones are this number here and this number here. What's step number 2? We're gonna need our double recombinants because we need those for step 3, which is middle gene ID. So what are our double recombinants? The smallest one, because recombination is rare and double recombination is a much more rare event. So 3 and 2 represent the smallest things on our list of things here. Again, to identify the middle gene, we need to compare the parents to the doubles. So we have 3 possibilities: either B is in the middle, G is in the middle, or R is in the middle. Take a second to see who thinks what. Take a vote. Okay, who thinks that B is in the middle? One person. 1 is the loneliest number. Who thinks G is the one in the middle? So many of you. Who thinks R is the 1? So everybody thought G. Everyone. Was I just a softie on this test? Let's see. Alright, we compare our parents to our doubles. We're looking for something changing. And if we do the analysis, these two look very similar because they both have a capital B and a little r, indicating that G should be the G in the middle. And these two are very similar because they both have a little b and a big R, big G in the middle. Look, apparently G is in the middle. Who would have thought? That's a bad choice on my part, apparently. Okay, we've identified G as the middle gene. We're gonna have a little map here of B to G to R. You can sort of skip the next step, which is rewrite order. They're already in the right order, but still want to remember we need to make sure that we write these genes in the proper order before going forward. What's the next step? So we're gonna do our single crossover. Between 1 and 2. Is anybody familiar with shorthand? If not, this little symbol means and. And this is between. You can also use it within, of course, as well. W and N. Shorthand's kind of fun, but I don't know all the shorthand. In music, we use the and symbol quite a lot because we count things. Anyways, which of our single recombinant offspring numbers go with the B to G crossing over event? So we take our parents, which are this guy and this guy, do the crossing over between them. You draw the crossing over right there. So what do we get as a result? Little b, little g, little r. Big B, big G, big R. So that's this one here, we'll call it S1 for single one. Where's the other one? Right here. Single one. Must have been feeling sorry for my class last year. You gave me, didn't make it easy. But they may not follow why these are this one here. Does they have the distance calculated already? Are you sure? Are you guessing?
That's what I've written down. If I was correct, I'm sure.
Okay, well, we'll see in a second, because I can look the answer up. That's what I got. And then the next distance, we already know what the next ones are. It's gonna be this one here as the other single. This one here is our other single. Which means we're gonna use this other crossing over event between G and R. And so we get a little b, a big G, a little r, which is the top one right there. And then big B, little g, big R, wait, that was the top one. And then, so little b, big G, little r, that's that right there. And we do the calculation for this distance. What do we mean? $50 plus 55 plus 3 plus 2. It's 105 plus 5 is 110. Divided by whatever the total is. I don't know what the total is. What's the total? What's the answer?
58.
What's the answer?
Got 11 point.
11.5. Hopefully that makes sense. That when you use 120 and 125, you get a bigger distance than when you use 50 and 55. And now between B and R, which is this distance here, gonna add those two things up. Did we get the right answer? Oh wait, I don't have the answers right here. I'll have to go here. 26.1, 11.5. Good job, everybody.
Could you like zoom in on it so I can take a picture of it?
This one right here?
I mean, either one, I just need like little numbers that are in the box.
That's stuff written on the board.
That's good, I just didn't see that part.
But we look at the review PDF, it's gonna sort of do the same type of thing of walk you through comparing the parents to the doubles, identifying the middle, and doing their combination, getting the products, and doing the calculation, and doing this, and getting the answer, and doing the next crossing over, getting the answer, and adding blocks. So this is the format I will use. Well, it's the format you'll see when you look at the walkthrough, or walkthrough. It's not called walkthrough.